हाँ जी कोई चलने से है. So today we are going to start another topic about of this chapter, and this is about the internal structures. When we talk about some internal structures of the prokaryotic cell, they may be of different types, and they may be of that may be a long list, starting from the chromosome, plasmid, cytoskeleton, storage granules, ribosomes, and endospores. So here, when we talk about the internal structures, it definitely means that the structures which are present inside the cell, or you can say that the structures which are present inside the cytoplasmic membrane. Because so far, up to this point, we have talked about the cellular membrane, cellular wall, capsules, slime layer, as well as the protein appendages. So, अब थोड़ी इंटरनल स्ट्रक्चर्स की बात करेंगे, और इंटरनल स्ट्रक्चर पे जब हम बात करेंगे तो सॉरी. अच्छा, इसमें मैं कुछ ज़्यादा डिटेल में नहीं जाऊँगा. मुझे लगता है कि क्रोमोसोम, प्लाज्मिड, साइटोस्केलेटन आपने शायद सेल बायोलॉजी के रेफरेंस से काफ़ी डिटेल में पढ़ा होगा. आई वुड जस्ट गो थ्रू, ताकि आपको इसके एक आइडिया हो जाए प्रोकैरियोटिक पॉइंट ऑफ व्यू से, माइक्रोबायोलॉजी के पॉइंट ऑफ व्यू से. एंड कुछ चीज़ें मैं आपसे पूछता रहूँगा साथ साथ, फिर हम आगे मूव करेंगे. सो स्टार्टिंग फ्रॉम द क्रोमोसोम, जब हम इसमें बात करते हैं तो वी आर फेमिलियर विद इट दैट दिस इज अ प्रोकैरियोटिक सेल, सो देयर वुड बी नो डिस्टिंक्ट nucleus. If there is no distinct nucleus, then where is the DNA and where is the genetic material that is present? So the genetic material of the prokaryotic cell that is present in center, or in in at a center of the cell, and this portion is just a gel-like region, and this portion is called nucleide. So keep in mind this gel-like region in the center of the cell that is not enveloped by any membrane. This is a gel-like region, just thicker than the other cytoplasm, and this is termed as the nucleide. In other words, we can say that this is the irregular mass. It does not have any distinct shape. It has a irregular, and it has a more viscous appearance, just like a gel. Then we talk about, if we talk about more specifically the chromosome, we can say that the chromosome of the prokaryotic cell, this is a circular, single है, circular है, and it contains a double-stranded DNA. Unlikely to the viruses they may contain the DNA as well as RNA, and some viruses they may have only RNA and only DNA, etc. But the bacteria, they have DNA, and more precisely, this DNA is double-stranded, circular है और single है, just one chromosome. Now, about the packing of the chromosome, we know that the prokaryotic cell that is smaller than the eukaryotic cell, it means that this single chromosome that would be tightly packed into a very small space. And if I compare the packing of the chromosome here, in this case, if you look into it, ये वाली जो diagram है, this is the color enhanced image of the transmission electron microscope. और इसमें जो दरमियान में जो reddish area है, this is more viscous and gel-like. This is actually indicating the presence of the chromosome. And comparatively, in this picture, the DNA that has been extracted out. अब इस केस में हमने किया क्या कि we have treated the cell with some reagents, and cellular envelope was broken, and as a result the chromosome that was extracted out, or the DNA was extracted out. Now you can imagine कि exactly DNA की या chromosome का size क्या है, एंड ये साइज कितने स्मॉल पोर्शन में या कितने स्मॉल सेल में पैक हुआ है. सो वी कैन से दैट द टोटल वॉल्यूम ऑफ द सेल. तो इस पिक्चर में जो मैंने अभी आपको दिखाया है, अगर हम फिर से इसको देखें तो exactly or originally the chromosome that is very huge, or the DNA of the bacteria that is very huge, but that is being packed into 10% of the total of the bacterial cell. You can just have an imagination. And how this packing is possible? This packing is possible by the process of the coiling and the supercoiling. Bacterial cell, it used to coil and supercoil the DNA and the chromosome to make it happen, to make it to visible in a very precise space. So this was about the chromosome. हम इसकी बाकी details में बिल्कुल नहीं जा रहे because I think that would be beyond the topic of that chapter. इसके साथ साथ अगर हम आगे देखते हैं about the plasmid, now in the prokaryotic cell or in the bacterial cell, the DNA that is not only present in the form of the chromosome, this may be present on the form of some extra chromosomal particles. अब यहाँ पे एक टर्म इस्तेमाल होगी प्लाज्मिड. प्लाज्मिड की डेफिनेशन के अकॉर्डिंग अगर मैं इसको यूं प्रोनाउंस करूं, the plasmid are the extra chromosomal part of DNA that is circular, double stranded and supercoiled. I repeat, plasmids are the extra chromosomal DNA which are circular, double stranded and supercoiled. For example, if we have a cell here, यहाँ पे हमारे पास अगर एक बैक्टीरियल सेल था, इस सेल के अंदर यहाँ पे हमारे पास ये अगर डीएनए था लाइक हियर, तो क्रोमोसोम जो है दैट वुड बी प्रेजेंट लाइक हियर, इस तरह से. सॉरी, प्लाज्मिड वुड प्रेजेंट लाइक हियर. दिस वुड बी द प्लाज्मिड. तो इसमें हम देख सकते हैं कि प्लाज्मिड that is extra chromosomal. This is other than the chromosome. This is supercoiled. This is double-stranded DNA. And what about the size of the plasmid? Its size ranges from 0.1% to 10% of the size of the chromosome. यानी कि क्रोमोसोम से भी छोटा है. अब सवाल ये है कि क्या इतने स्मॉल साइज पे इस प्लाज्मिड में कोई स्पेसिफिक जीन्स प्रेजेंट हो सकती हैं या नहीं. इस प्लाज्मिड के ऊपर yes, definitely the genes are present and the genes may vary from few to several hundred. अब यहाँ पे इम्पोर्टेंट बात यह है कि प्लाज्मिड के ऊपर जो जीन्स हैं, the genes that are not the housekeeping genes. Housekeeping genes are the genes which are required by the cell for its basic function. So the housekeeping genes that are not present on the plasmid. Housekeeping genes are present only on the chromosome, and on the plasmid, the genes are present for extra characteristics. For example, you can say that the antibiotic resistance, the genes which are responsible for the antibiotic resistance, they are present on the plasmid. और सिर्फ इतनी सी बातें हैं कि एक प्लाज्मिड जो है ये एक्स्ट्रा क्रोमोसोमल है. दिस इज स्मॉलर देन द क्रोमोसोम. It does not have the housekeeping genes. It only have the advantageous genes, जैसा कि antibiotic resistance के genes हैं. और प्लाज्मिड की बेस पे ही फिर हम आगे conjugation को define करते हैं. जैसा अभी हमने पिलाई के केस में पढ़ा था कि एक सेक्स पाइलस बनता है, एक बैक्टीरिया का डीएनए दूसरे बैक्टीरिया में ट्रांसफर होता है. तो इस conjugation के प्रोसेस में प्लाज्मिड इंपॉर्टेंट रोल प्ले करता है. And there are some specific plasmid which are called F plasmid and F minus plasmid and F plasmids. The bacteria which have the F plasmid, they may be termed as the F positive. उसके बाद HFR strain बनती है. तो ये अगर कभी हमने कोई ऐसा सब्जेक्ट पढ़ा जिसमें माइक्रोबियल जेनेटिक्स इन्वॉल्व हुई, तो उसमें हम ये चीज़ें डिटेल के साथ पढ़ेंगे. लेकिन just to give you an idea that plasmids, they have important role in the conjugation. And what is the conjugation? Conjugation is the transfer of genetic material from one cell to the other cell. Okay, Ji. Up to this point, if you guys have any questions, you may ask.
No,
any
sir.
questions? Plasmid की समझ आ गई है? What is the plasmid? Okay, I suppose. Okay, the next internal structure that we have to cover is the ribosome. As we are familiar with it, that ribosomes, they are the protein synthesis factories, and their structure actually that facilitate the joining of the amino acid in a specific sequence. Now we have an idea that the ribosomes of the prokaryotic cells, they are of smaller size, and they are of only 70 s. Yeah. Just a minute, please. In the next class, unfortunately, मुझे डॉक्टर सुहेल के साथ एक मीटिंग में जाना है अभी फौरी तौर पे, so that's why I couldn't continue this. I cannot continue actually. मुझे अटेंडेंस दे दें.
Okay, sir.
फिर हम इसको नेक्स्ट क्लास में पढ़ेंगे. Yes, Afira Farouk. आकिफ बुरहान
Present,